Okay, hello to all of you. Let's discuss chapter number 7. And chapter number 7 basically focuses on DNA and DNA technology. So basically, I have posted many videos. I want all of you to watch those videos because those videos talk about many cases where people have been proved innocent or guilty on the basis of the DNA test. Basically, some people were in the prison because earlier forensic science was different. They used to, do things, do the analysis on the basis of either fingerprints or the hair analysis. So slowly, as science has developed, biology has developed, now we have a DNA technology. Now the forensic scientists use the technique called DNA fingerprinting or DNA profiling, which we'll be discussing in this chapter. So basically, this technique or this technology is very authentic, it's very genuine, and on the basis of that, the real culprits of the crime can be found. This technique works on some specific or the special regions or the unique regions in our DNA, which are unique to every individual. So this technique is also called DNA fingerprinting. So just like the fingerprints of an individual, every individual has unique fingerprints. So similarly, we have some regions in our DNA which are unique to every individual. So this technology basically works on, basically makes use of those unique regions in our DNA. So please watch those videos and you will understand you will get a lot of feel about what I'm talking about right now. Okay, so let's talk about what is DNA, what is structure, and how DNA replicates. So all these concepts are very important to understand this DNA technology. So basically DNA, as you know, that our genes, they're made of DNA. So what are genes? Genes are those entities which are passed from parents to the children. So all the characteristics we possess, like how we look, how we behave, what kind of diseases we are susceptible to. So all that information is there in our DNA. DNA is a genetic material. So inside our cells we have nucleus. In the nucleus we have chromosomes. Every species has a different number of chromosomes. Every species has a specific number of chromosomes. Like if we talk about humans have 23 pairs of chromosomes. So these chromosomes, they are made of DNA, and the DNA is wrapped over the proteins. So on these chromosomes are the genes, okay, and these genes are responsible for all the characteristics we possess, and these are the genes are the ones which are passed from generation to generation, and these genes are made of DNA wrapped over some proteins, and those proteins are called histone proteins. So DNA, its full form is deoxyribonucleic acid. So it's a hereditary molecule that's passed from parents to the offsprings. I have already explained what basically DNA is. Okay, so where DNA is, it's in the nucleus of the eukaryotic cells, because prokaryotic cells, they do not have the nucleus, but they do have genetic material. But their genetic material, which is in the form of a single chromosome, is basically sitting in the cytoplasm, and that region is called the nucleoid region. So prokaryotic organisms like bacteria and archaea, they do not have the nucleus, but they do have the genetic material. So DNA is in the form of chromosomes, and the DNA is wrapped around the proteins, as I said. So you can see here, let's actually go in this picture. Hold on. Okay, so this is the cell, and inside the cell there is a nucleus, and in the nucleus are our chromosomes. So you can see these are the chromosomes, and these chromosomes are basically made of DNA. So this is the DNA, and they are wrapped over histone proteins. So these are the proteins, and this DNA is wrapped very, very tightly over them. So as I said, every species has a specific number of chromosomes. Like humans, they have 23 pairs of chromosomes. Humans have 23 pairs of chromosomes. It's very important to understand this concept that the chromosomes, they are in pairs. So in every pair, which has two chromosomes, one chromosome comes from the mother, other chromosome comes from the father. As humans have 23 chromosomes, as humans have 23 pairs of chromosomes, out of these 23 pairs, 22 pairs of chromosomes, they are called autosomes, and the 23rd pair is called the sex chromosomes, and this pair determines the sex of an individual. If the 23rd pair is both XX, then the individual is a female. If it is XY, then the individual is a male. Every person's DNA is unique, and that determines all the characteristics we possess. So let's talk about the structure of DNA now. Okay, so basically we have already talked about in chapter number 2 when we're talking about the macromolecules, that nucleic acids, there are two kinds of nucleic acids, DNA and RNA, and they're polymers, and they are made of monomer called nucleotide. So the basic subunit of DNA is a nucleotide. So now let's talk about the structure of a nucleotide. Now every nucleotide has three components. One is a sugar. Okay, it's a pentose sugar. Why it's called pentose sugar? Because it has 5 carbons. So you can see this is the pentose sugar, carbon number 1, 2, 3, 4. Sorry, carbon number. So these are the this is the pentose sugar, and this is carbon number. So there are 5 carbons in this. Carbon number 1, 2, 3, 4, and 5th one. No, I'm sorry. Actually, here is the oxygen. Here is the oxygen. This is carbon number 1. This is carbon number 1. This is 2. This is 3. Here is 4. On 4th one is attached the 5th carbon, and to the 5th carbon is attached the phosphate group. Here is the oxygen. I'm sorry. So this is a pentose sugar. Okay, and here is attached the nitrogenous base, and here is attached the phosphate group. Now this phosphate group has a negative charge, and because of the negative charge on the phosphate group, DNA possesses negative charge. So it's very important to know that DNA has negative charge on it, and at this position is attached the nitrogenous base. Now what are nitrogenous bases? There are nitrogenous bases are basically ringed structures which have carbon and nitrogen in them. That's why they're called nitrogenous bases, and their pH is alkaline, so that's why they are called nitrogenous bases, and they are attached here. There are 4 kinds of nitrogenous bases: A, T, C, and G: adenine, thymine, cytosine, and guanine. So let's talk about. So as I said, DNA is composed of nucleotides. So nucleotide is the basic subunit or a monomer of DNA, and every nucleotide consists of a sugar, phosphate group, and a base. So you can see here, this is the pentose sugar, this is nitrogenous base, and here is the phosphate group. And these nitrogenous bases, there are 4 kinds of nitrogenous bases in DNA: A, T, C, and G: adenine, thymine, cytosine, and guanine. So these are the 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. Now the DNA is double stranded, which means it has 2 strands. So you can see here, this is one nucleotide, and these nucleotides, they're all linked with each other because DNA is a polymer. So polymer is made of monomers, and these monomers, they're all linked with each other by covalent bonds. But DNA is double stranded, so which means that there are 2 strands of DNA, and these 2 strands of DNA, they are linked with each other, and they're linked with each other by very light weak bonds. They're called hydrogen bonds. So the 2 strands of DNA, they are linked, they are connected to each other by hydrogen bonds, and these nitrogenous bases, they link with each other by complementary base pairing rules. And on the basis of these complementary base pairing rules, A always links with T and C always links with G. A always links with T and C always links with G. So the 2 strands of DNA are held together by base pairing, hydrogen bonding between bases, and they are linked, the two strands are linked with each other on the basis of complementary base pairing rules. A always links with T and C always links with G. And basically, if this is one strand of DNA, okay, and this is second strand of DNA, these two strands are linked with each other. So in each strand, if you see, these are the nucleotides, and now every nucleotide has a sugar, phosphate group, and a base. Now these bases are of four kinds. So if you look at this one strand of DNA, every nucleotide has a sugar, has a phosphate group, but what is different is the nitrogenous base. Okay, so you can see here is C, here is T, here is A. So in every nucleotide, the sugar is same, phosphate is same, but what is different is the nitrogenous base. So the order of these nitrogenous bases is called the sequence of DNA. So if A is coming first, or T is coming first, or C is coming, so it's the order of these nitrogenous bases. So the order of these nitrogenous bases is called the DNA sequence. Okay, so basically, so as I said, like for DNA fingerprinting technique or DNA profiling technique, you will see in those videos that how this technique has helped many cases. So for that, first of all, suppose there is some crime happened at some place, and these forensic scientists go there, or the police goes there. So what are they going to look for? They're going to look for some samples. They will look for either blood sample, or they're going to look for blood sample, saliva sample, or a semen sample, right? So from these samples, what are they going to do? From these samples, they are going to actually extract the DNA. Now the sample will be so less there, so it is like I mean, if you just go to a crime scene and you just get a drop of blood, and from that drop of blood or from a saliva sample, you just get one drop. From that drop, if you have to extract DNA, you are going to end up with a very little amount of DNA, right? So with that little amount of DNA, you cannot do the analysis. So what has to be done? So that DNA has to be replicated. Okay, so basically there is a technique called PCR, which we call polymerase chain reaction, by which we can amplify the DNA, in the lab. Okay, so PCR is a part of the DNA profiling technique. Before we go and discuss PCR, I'm going to discuss how actually DNA is replicated inside the cells, because it's the same technique, or it's the same principle which is used or which is applied to perform the PCR. So PCR is a in vitro technique. So let's discuss that. So basically, DNA replication, it works on a principle. It works on a principle, or it multiplies. DNA replicates or duplicates, not multiplies, it's wrong terminology. No multiplication. It's basically duplication or amplification of DNA. It takes place, it's called the semi-conservative mode. So basically what happens is, as you know, that DNA has two strands. These two strands actually separate, and then every strand of DNA is going to act as a parent strand or a template strand, and a daughter strand is going to be made on the basis of the complementary base pairing rules. So you can see here, like this is your double-stranded DNA. First of all, these two strands, they need to be separated. So for the separation of the two strands, an enzyme is required inside our cells, and that enzyme is called helicase. And majority of our enzymes are protein in nature. So helicase is a protein. So this is your double-stranded DNA. First of all, the two strands, they need to be separated, and that happens with the help of the enzyme called helicase. Now the two strands have separated. Both strands act as parent strands, or we can call them, they are a template strand. So parent strand or a template strand means that those strands, they have a sequence in them. Sequence means they have an order of nitrogenous bases, A, T, C, G. Okay, so now whatever is the sequence on the parent strand, a complementary daughter strand will be made with the help of the enzyme called DNA polymerase. Okay, so the two strands separate, then the two strands act as parent strands, which means whatever is the order of the nitrogenous bases there, a complementary daughter strand will be formed, and as a result of that, two molecules of DNA are produced, and those two molecules of DNA, they are identical to each other as well as they are identical to the parent molecule. So this process basically involves few enzymes and some other proteins, but we are not going to discuss in a lot of detail with you guys, so we are keeping it simple. So with the help of the enzyme helicase, the two strands separate, and with the help of the enzyme called DNA polymerase, a daughter strand is made, and the daughter strand is made on the basis of the sequence which is there on the template strand. So this is called semi-conservative mode of replication, because when from one molecule of DNA, so you can see this is one molecule of DNA and two molecules of DNA are formed. So you can see the two strands which are there in the old molecule of DNA, they are shown with blue color, and the new daughter strands, they are shown with green color. So from one molecule, two molecules of DNA are formed, and these two molecules of DNA, you can see that they have one strand, they have blue, and one strand is green. So one strand is old, one is new. So that is why this mode of replication is called semi-conservative mode of replication, because from one molecule of DNA, when two molecules of DNA, they are formed, one strand is old strand and one strand is new strand. So that is why it's called semi-conservative mode of replication. So this is all what we discussed. Okay, now we are going to discuss the technique called polymerase chain reaction, and this is a technique discovered by scientist called Carey Mullis, who recently died, and he actually found this technique. Basically, the principle of this technique is the principle is basically how the cell, how the DNA actually replicates or duplicates inside our cells. Okay, the principle is same, but this is a laboratory technique. This is done in the test tube. So basically, let me overall explain you what polymerase chain reaction means. So basically, like you can have, if suppose you have a very small amount of DNA, but for the analysis you need large amount of DNA. So in this technique, you can take your template DNA, whatever the DNA you want to amplify. Now, if you have a template DNA and it's a very long DNA, you don't want to replicate everything, right? You want to replicate a specific part on the, if you want to replicate a specific part or the region of the DNA, then you have to use primers. Now, what are primers? They are short segments of DNA that can guide DNA polymerase to the section of DNA to copy. So primers, they're short stretches of DNA. So primers, you can get them synthesized from some companies. So primers are short stretches of DNA, and they can go and cover those regions of the DNA, those specific regions of the DNA which you want to amplify, because their sequence is such that they will go and they will cover those specific regions of the DNA, and they're chemically synthesized by the companies. So what you need to when you want to replicate the DNA in the lab, you need a test tube. You add your template DNA, you add the primers, you have to add DNA polymerase, and then you have to add nucleotides. And then there's a machine. It has some slots. You put the test tube, like this is your microcentrifuge tube. You put your tube on that machine, and the machine is connected with the computer, and the computer has a software, and basically the components which are there in the tube, they go through some cycles, and every cycle has some steps. So it's all up to you how much DNA you want to amplify. If you want to amplify only, basically you need only few nanograms or micrograms, you can set for 20 cycles or 30 cycles. If you want the DNA in milligrams, you can set to like 60 cycles or 70 cycles. It all depends on your need. and in it all depends on your need, how much DNA you need, and every cycle, so it's all up to you. You have to feed in the software how many cycles you need. Every cycle goes through some steps. First step is heating, or we also call it a denaturation. So the components which are there in the tube, they are going to go through a very high temperature, which is around 94, which is a very high temperature, and that denaturation temperature is going to separate the two strands of DNA. Okay, and then let me explain you here. So first of all, the two strands of DNA will separate, and then the primers are going to, okay, so like this is your template DNA. These two strands will separate, and suppose you want to like amplify this much part of the DNA. The primers are going to go and bind here. One primer will go bind this side, another primer will go bind this side, and then the second step in the PCR is called annealing. Annealing is basically when the primers, they go and attach, and third is elongation, in which the nucleotides are going to go and bind to the primers, and the strand will be elongated. So denaturation, annealing, and elongation. So this is the way from one molecule of DNA, two molecules of DNA will be formed. So this is first cycle or first round. Okay, and then in the second cycle, this one molecule will form two molecules. This will also form two molecules. So from 1, 2 molecules of DNA will be formed, from 2, 4 will be formed, and from 4, 8 molecules of DNA will be formed. So that's why I said how much DNA you need. On the basis of that, you have to set up the cycles. So this is basically PCR. So this is a technique which is done in the lab by which we can amplify the DNA. So this is all what I explained. Now let's talk about the technique called DNA profiling, or it's also called DNA fingerprinting. So basically in this technique basically makes use of some special regions in our DNA which are unique to every individual. So that's why this technique is called DNA fingerprinting. As every individual has unique fingerprints, so similarly in every individual's DNA there are some regions which are unique to that individual, and those regions are called STRs. They're called short tandem repeats. Okay, so in this technique those regions are basically used, and they can help us in solving various problems of crime or finding who is the biological father, all these kind of things. So those regions which I talked about, which are unique to every individual, they're called short tandem repeats. They're called short tandem repeats. And PCR, the technique which I just discussed with you, this is a technique by which this is a technique by which we can amplify the DNA. So this technique is used to amplify the STRs, because otherwise they will be because their amount is very less, we cannot do the analysis. So we need to replicate those, we need to amplify those regions, only then we can do the analysis. Okay, so this is basically showing you what STRs are. So STRs are sections of chromosome in which DNA sequences are repeated. So they're called short tandem repeats. Okay, so these regions, like you can see here, for example, so they're called short tandem repeats. So these are some short sequences. So these are some short sequences which are repeated. Okay, like you can see here, this is this is an example of a sequence called AGCT. Here is an example of a sequence called AGCT, and this is this, like in every individual, this sequence can be repeated in a different number of times. Like you can see, so this is the this is individual number 1, this is individual number 2, this is individual number 3. So as our chromosomes, they are in pairs. So this is the maternal chromosome, this is paternal chromosome. You can see that on this individual number 1, this STR, this AGCT is basically, this STR is repeated 6 times on the maternal chromosome. It is repeated 4 times on the paternal chromosome. In individual number 2, This STR is repeated 3 times on the maternal chromosome and 6 times on the. So basically, there are different kinds of STRs. There's not only AGCT, there can be GCC, CGG, it can be AAA, ATT, it can be different numbers. But the important thing is these are repeats, they are repetitive sequences. These are repetitive sequences and they are repeated they are repeated in a different way in different individuals, and that's make these regions unique in every individual, and that is what is made use. This is what is used in this technique called DNA profiling. Exact length of STR varies from person to person. So this is what I just explained. So basically what is done is when you, like here you can see these are the cheek cells in the saliva. So at the crime scene, if somebody found these cheek cells in the saliva, first of all the DNA will be extracted. The DNA will be extracted, and then these STRs, they are going to be amplified by PCR. So they are amplified by PCR, and you can see that this is like STR from region number 1, this is STR from region number 2, this is STR from the region number 3. So first of all the DNA is extracted, and then these STRs, they are amplified by PCR, and then the different samples from different individuals, they can be run on the DNA gel. Now DNA gel are usually made of agarose, and in the agarose gels there are pores through which the DNA sample is going to move, and it moves with the help of electric current, and this technique is called gel electrophoresis. So we have in our labs we have agarose gels, and in these agarose gels we have like different compartments like this. We call them wells, and this agarose is a jelly kind of a material which is solid at room temperature, and there are pores inside, and there are pores inside, and the DNA and we apply the electric current. As DNA is negatively charged, so it is going to move towards the positive electrode. So the DNA moves through these pores, and it gets separated, and the separation of the DNA fragments is on the basis of the size. The DNA fragments which are very big in size, they move slowly through the gel, so they stay close to the well, and the DNA fragments which are very small in size, so the DNA fragments which are very small in size, they move away from the well. Okay, so they travel more distance. So the short fragments, because they're small, they can move easily through the pores of the gel, they move away from the well, and the big fragments, they stay close to the well. So this is the way what we do. What is done in this technique is, so you can have a sample from the crime scene, and you can have like these are different suspects, so you can extract their DNA and you can amplify the STRs, and then you can load their DNA in these different wells. So you have this is a DNA sample from your crime scene, this is from different individuals, so the STRs are all amplified, and you run on the gel, and you can see how these DNA fragments, they are separating. So the big fragments, they stay close to the well, and the small fragments, they move away from the well. So basically what is done in this technique is the pattern of these DNA fragments is observed, and the DNA fragment pattern after running on the gel is compared with the DNA fragment pattern of the sample from the crime scene. So this is the DNA fragment pattern. This is a DNA fragment pattern from the crime scene, and this is the DNA fragment pattern from different individuals, and it is going to be matched. And basically, so suppose like this is your the saliva sample from the crime scene, and these are 3 different individuals, and you can see here you have to match the DNA fragment pattern from the different suspects. You have to compare it with the crime scene, and you can see here we have 3 suspects, suspect A, B and C. It is the suspect C's DNA fragment pattern which is exactly matching with the sample from the crime scene. So this is the culprit because it exactly matches. So you can see this band match with this band match, and this band match with this with this, red one this, blue with this. So this is the way how the paternity problems and the who is the criminal or something, all these situations there. And this is called DNA fingerprinting. Okay, so this was DNA fingerprinting. So as this is a chapter basically on genes, now I am going to relate the genes, the chapter on genes with cancer biology. So in the cancer biology, and I've also posted many videos for cancer genes. So there are two very important genes in cancer. They're called oncogenes and they're called tumor suppressor genes. So oncogenes are those genes basically which drive the cancer. So they are the drivers of cancer, which means that they make cells become cancerous. And tumor suppressor genes are those genes which basically stop the growth of the cancer cells. So oncogenes are those genes which make the cells cancerous, and tumor suppressor genes are those genes which actually stop the growth of the cancer cells. So oncogenes, to understand oncogenes are actually those genes which are actually present in our cells, and they are the ones which control the cell division. So they are the normal genes. So you must have heard sometimes some people say we all have cancer genes, right? So basically we all have genes which are called proto-oncogenes. Okay, so we all have genes called proto-oncogenes, and these proto-oncogenes are those genes, which are involved in the cell division. Okay, so they are the ones which are making our cells divide in an organized manner. So what happens in cancer is these proto-oncogenes become oncogenes. They turn into cancerous genes. So how does that happen? It happens because of mutations. So proto-oncogenes, which are normal genes, they get mutated, they get changed. So what is a mutation? It's a sudden change in the DNA. And because of the mutations, these proto-oncogenes become oncogenes, and they make normal cells become cancer cells. And now what are tumor suppressor genes? So tumor suppressor genes are those genes which are basically guardians of our genome. They are the ones which are like check if everything is going correctly in our cells with the cell division, if our cells are dividing in a correct way. So if there is something wrong in the cell division, the function of the tumor suppressor genes is to suppress the growth of the cells. So the function of tumor suppressor genes, is to stop the growth of the cancer, growth of the abnormal cells. So if there is something happens in our cells and our cells starts becoming abnormal, the function of tumor suppressor genes is not to let those bad cells to grow. They will stop, or they will have a check on the growth of the bad cells. That is what is the function of tumor suppressor genes. But what happens in cancer is these tumor suppressor genes, which are the good genes, they also get mutated. And because of their mutation, what happens is, because of the mutation, they cannot function properly. Okay, so tumor suppressor genes are those genes which are like a guardian of our genome. They make a check if everything is going in a correct way in our cells. But what happens in cancer is these tumor suppressor genes, they get changed, they get mutated, and as a result of that, they do not function. So now I'm going to show you, or I'm going to discuss with you some of the events which can take place in our genes, because of which the normal genes become mutated or they get changed, and as a result of that what happens is our cells starts growing abnormally. So basically cancer is a disease in which the DNA gets mutated, and in next chapter we will understand that the DNA which is making our genes, it is actually some information. It is just some information. So this information which is there in DNA is basically coding for proteins, because proteins are the real players. They are the ones which are performing all the functions in our cells, majority of the functions in our cells. So because of mutations or some changes taking place in our DNA, because of that proteins get changed, proteins get mutated, and these proteins, they are involved in the cell division. So when DNA gets changed, protein gets changed. When protein gets changed, these proteins are involved in cell division. So when proteins get changed, our cell starts dividing abnormally, and the abnormal division of the cells is the disease called cancer. So here basically what I'm talking about is, so this is the DNA, this is the proto-oncogene, basically it's shown here. So this shows that how does, so this slide is basically showing how proto-oncogenes can be converted into oncogenes through several distinct mechanisms. So basically, there can be a point mutation, which means there can be a sudden change in the DNA, and that change in the DNA is change in the nitrogenous base. Suppose in a gene there is adenine, it gets changed to guanine, or if it is thymine, it gets converted into cytosine. So there can be point mutations, okay, because of which because of the point mutation, there can be the formation of a abnormal hyperactive protein, and there can be gene amplification, which means some of the genes can get amplified, and because of that the protein becomes in excess. The protein will be normal, but it will be in excess. That's because of gene amplification. Or there can be various things can happen in our genes, chromosomal translocations, which means basically means, like you can see here, this part of the gene goes and joins this part. So now the gene is changed. Okay, so various things. So point mutations can take place in our genes, gene amplification can take place in our genes, chromosomal translocations can take place in our genes, local DNA arrangements, like there can be some insertions, there can be some deletions, there can be inversions. All these events can lead to the formation of abnormal hyperactive proteins. Or even in our genomes, some of the viral DNA, can get integrated, okay, and that can result in the formation of excess normal protein. So what happens is, because of the changes in our DNA, that can be either a mutation, gene amplification, chromosomal translocations, local DNA rearrangements, or it can be some insertional mutagenesis. Because of all these events taking place in the DNA, it can result in the production of the protein, which is in excess amounts. It can result in the overexpression of the proteins. It can result in the overexpression of the proteins, or it can result in the formation of abnormal hyperactive proteins. Okay, so this is what is the crux of this chapter. So we talked about genes. In the next chapter, we are going to talk about how does the information in the DNA gets coded into the proteins. If you have any question, please let me know. You can send